start recording. Right, share screen. Is my screen visible?
Yes, ma'am.
Yeah, okay. So, sure. So the number of people who join is going on decreasing. So, anyway, this is the sixth session. We have six more sessions to go. We are halfway through the course, and I'm again back in Bikaner. I'm hoping that the power stays fine and the Wi-Fi stays fine till the end of the class. Yeah, looks like I'm going to be traveling a lot before this course actually ends. But yeah, I'm hoping to finish whatever I started. So in the past few sessions, we've been looking at different ways to classify microbes, different methods that are involved. We looked at transcriptomic, proteomic methods, genomic, metagenomic, metabolomics, and so on and so forth. Today also it's going to be something related to the methods itself. How we can identify them and then how we can classify them, uh, and what are all the different techniques involved and so on. Yeah, so the slides are designed in such a way that you can answer the questions in your assignment. So I hope so that so far whatever slides I've been having is helpful in answering the questions that are asked in your assignments. Again, to zoom out and look at the bigger picture, ecosystem has every organism, every possible organism, along with the abiotic factors in that particular environment. That is what makes an ecosystem. And ecosystems are very complex and they are also very dynamic. Dynamic in the case that they keep changing, they have changed a lot throughout the course of evolutionary history. They keep changing from day to day, and even within the same day there's a lot of changes that happen in the abiotic factors, as in it may rain, and the rain may change the microhabitat a little bit, that some organisms might come out after it rains, some might not come out after it rains, and so on. So yeah, ecosystem keeps changing, and hence it's a dynamic complex because of these different interactions with the abiotic factors and the biotic factors. And a particular ecosystem could be made up of multiple habitats. Here in this image we can see that there is a pond, and there's this rocky area like structure, and then there are grasses and trees and so on. So a single ecosystem could have multiple habitats, could have an aquatic habitat, could have a dry habitat, could have a green trees and so on. So the ecosystem could change in these habitats, and hence the organisms that inhabit these different habitats could also be different within a given ecosystem, and this encompasses all the microorganisms too. So the microorganisms that we find in different habitats in a given ecosystem could be very different from one another. So how do we understand this diversity and the species richness and so on? It could be just directly from the field observations, to just observe what are all the different microbial processes that are happening, what are all the different habitats, and what could facilitate the growth of different microbes in these different habitats. It could be just that kind of observations from the field that are done along with proper statistical methods. Or it could be from laboratory testing, where we systematically test the presence of different microbes using different standard methods and protocols in the laboratory. And often, what people do in microbial analysis, where people study microbial communities in a given ecosystem, is by following a combined technique where they combine field observations, information from the field, along with the information that we get from the lab tests and the lab work. Apart from the field observation and lab environment, which involves actually studying the microbes, one can also do mathematical testing of hypothesis, where we can just model the different components of an ecosystem, like abiotic components, other living organisms, plants, and see how the microbial diversity could change and the abundance and species richness could change just by substituting these mathematical values or empirical values in equations and testing a hypothesis. And this particularly works in case of environments or ecosystems which are broadly under characterized or very poorly understood, regions that we do not have much information about, both from field observation and laboratory testing, could be mathematically modeled to predict what kind of diversity and what extent of diversity could be supported using mathematical models. So in this session, or at least for this course, we'll mostly only be talking about the laboratory testing methods, how we can isolate microbes from the environment and classify them and characterize them for our purposes or for the application that we need them for. So the microbial cultures isolated from environment need to be classified. That is, they need to be identified and taxonomically placed along with the rest of the microbes that are found in that particular habitat, and characterized in a way that we understand their metabolism, their ecology, and physiology, so that we can use them for whatever application we want to use them for. So, first thing is, how do we classify them, or what are all the different methods that can be employed to classify microbes? The first and the foremost thing that we need to understand while working with environmental samples, may it be water samples or soil samples or any of the environmental samples, is that there could be thousands and thousands of microbes present in that given environment, but not all of them can be cultured in a lab. So for any microbe, this is how the microbial growth phase works. There is a lag phase. So we, let's say we take the microbes from the environment and then we culture them in a suitable media. So how do we determine the suitable media? We just have to try out different media with different sugar components. So in the previous sessions, we saw what are all the different components that are required for the metabolism of a microbe. So they need sugars for carbon source, they need an energy source, they need an electron transport source. So these are the different things that we should ensure that is present in the microbial culture, so that they can grow suitably. So a microbe that is cultured in the environment has these different phases. The first is a lag phase, where they do not start growing yet. They are just getting acquainted to the environment. So we're shifting them from their original environment to the lab environment in a laboratory culture. So they have to first get accustomed to that sugar molecule, that energy source that we are giving. So that phase where there is no growth evidently observed, but the microbes are alive, is called as a lag phase. And then there is an exponential phase where the microbes exponentially start growing. So here, that is what is depicted in these images too, where there are favorable conditions. Favorable conditions indicate both presence of food resources, the microhabitat, the environment. So microbes, at least when they're cultured in the lab, need to be taken care of to make sure the humidity conditions are right, the temperature conditions are right, and whether there are other microbes that are inhibiting the growth of the particular microbe of our interest, and so on. So those come under favorable conditions. So under the favorable conditions being present, we can see that the microbes will shift from a lag phase to an exponential phase, where the growth increases exponentially. So we how do we call this exponential? Because when we plot the number of cells that are present in a given volume of cell culture, we will be able to make this graph, and if we put a mathematical equation to this graph, we'll see that the numbers are changing in an exponential scale. So it could be changing on a linear scale, logarithmic scale, and exponential scale. So these are the three scales in which we generally measure anything, and we see that microbes, when they start growing exponentially. So one becomes two, two becomes four, four becomes 16, and so on. That's how an exponential scale goes. So the microbes start increasing in an exponential phase, and then followed by a stationary phase. So what happens after the exponential phase is that now they are actively utilizing all the energy and they're growing. That's how the exponential phase comes. But later on, after some point, they start dying as well, because there's not enough amount of energy present for the microbes to just keep growing, to just keep increasing in number. So they consume the energy, but some of them also start dying because there's not enough energy source and sugar source available. But the number of cells that are alive and the number of cells that are dying is equal, and hence we see that the overall curve remains the overall curve remains stationary. The curve remains like a flat line. So it's called as a stationary phase. Just a second. Yeah, it is recording. Anyway, right. I'm still audible, right? There's no issues hearing me. Can someone just confirm?
Yes, ma'am.
Yeah, okay, thank you. Yeah, so that's
Audible.
the sorry? Yeah,
No,
yeah.
you are audible.
Yes, okay, thank you. Yeah, so that's the stationary phase. And during this stationary phase, they can undergo some kind of stress or adverse conditions in terms of, like I mentioned, it could be because of less availability of energy sources or of sugar sources, and that could put them in stress, and they might start going into a dormant phase. So this phase of deep dormancy, where there is an inactivation of growth, after this point they can either start regrowing again. They can get back to this viable phase and start growing back again, or just stop growing for a while. And when conditions become good, available, or permissive conditions start appearing again, they can get resuscitated and go back into this growth phase of exponential phase and so on. This happens after they enter the dormant phase. But that's not what happens for all types of cells. All microbes can't be resuscitated or start regrowing. Some of them permanently die, permanent cessation of life processes, that is, cell death happens in some of the cases. So this all of this process is for a bacteria or microbe that is culturable in the lab, that can take up sugar or energy from the laboratory sources and can grow under laboratory conditions. But there are n number of microbes that we will find in the environmental samples that are not culturable. They may be in the deep dormancy phase because we're collecting them from their natural environment where they're actively growing. They may grow into a deep dormancy phase, but may ever like they may never revive back again or resuscitate again to start growing under the laboratory conditions. They may be able to divide. They can be cultured under different conditions, but yet it is just not understood how to culture them under laboratory conditions. So if you take them back to the environment, they may start growing. So they are able to divide, but some of them might not be able to divide at all because they are in the form of spores or resting dormant cells that do not grow until they start finding the environment suitable for growing. So there could be these two types of unculturable cells that or unculturable microbes in the environmental samples. So this is what is a visual representation of what might happen when we collect environmental cells. So we are collecting environmental samples, and we can observe them under the microscope and we can see n number of microbes. But when we actually start culturing them, we'll see that only a very small proportion of the total number of cells are actually culturable, and a huge lot of them that might not even be microscopically observable are actually something that can't be cultured in the lab. So this is just like, a tip of the iceberg is what we know about how to actually culture them and grow them in the laboratory conditions. And there's a huge mountain of information available with the unculturable cells that we do not know how to culture or how to grow. So this is called as the Great Plate Anomaly, where whatever we think, whatever we have achieved about understanding them in the lab is very little compared to what is actually out there. So if we actually put a percentage to it, we know only about 0.1 to 1% of bacteria and how to grow them or culture them under laboratory conditions. So that's the Great Plate Anomaly. So, in such conditions where we do not know how to culture a lot of microbes, what do we do? How do we actually understand what is out there, understand their diversity? How do we classify them, and so on? Since several species of bacterial organisms or microbes cannot be cultured in the lab, it would be difficult to find a suitable media to grow them and then isolate DNA, because in the previous sections as well we have seen that we need to culture the cells and then collect all of them, isolate DNA, and then amplify specific regions using PCR, so that later on we can see genetically what strain or what organism they are. So this is how any conventional uh, method of classification or phylogenetic assessment works for any organism, not just microbes. But in cases where we can't even culture them, we can't even know how to culture or what suitable material to use for culturing them, there is no question of isolating DNA and carrying out a PCR. So in that case, ideal method for identifying all of the bacterial or microbial organisms that are present is to do a metagenomic analysis, where we can sequence a reliable marker such as the 16S rRNA. So 16S rRNA is a ribosomal rRNA subunit. So the ribosomes are where the proteins are actively produced, and these ribosomes are made up of RNAs. So this is also one of the reason why RNA is considered as the most basal biological molecule that came into existence, because many of the microbes and viruses have RNA based genetic information, and RNA also forms the basic ribosomal subunits, which is required for making all the proteins that are there in our body. So any cell which has any protein that has developed comes from the rRNA, from the ribosomes. So 16S is one of the subunits for the microbial ribosomes. So this is a component of a small subunit of the prokaryotic ribosome, and this sequence is often used as a housekeeping genetic marker. So what's an housekeeping gene? So housekeeping gene is a gene that is universally present in all the cells of a particular organism and expressed under all the conditions. So in case we are looking at expression of a particular cell, or if you're trying to sequence a particular cell and to know whether whatever the sequencing reagents that we have used and whatever the sequencing process that we have carried out have actually successfully worked, and it's not because of any human error or because of any reagent error, we always use a housekeeping gene, which is usually produced in abundance and available in abundance, and hence can be sequenced under all conditions. So 16S rRNA is one such gene, which is a housekeeping gene, and it's a marker which can be sequenced for identification and classification of bacterial species. Why is this so ubiquitous? Why is this so universal? Why is it such a standard housekeeping gene? Because this is, like I said, it's present in almost all bacterial species, and ribosomal rRNA, different subunits are present in higher order organisms as well. So not just 16S rRNA, other rRNA subunits are also used for such housekeeping gene sequencing. So 16S RNA is there in almost all of the bacterial species of interest that we are trying to understand. All right. And the sequence is highly conserved because ribosome, like I mentioned, is the most basic unit in any cell, which is important for protein production, it should be there in all the cells, and changing any of the important sequences or any of the important amino acids that are responsible for this whole ribosomal mechanism is altered, the whole cell and the whole organism will collapse. Hence this gene is highly conserved across different bacterial organisms, but there are some sites which do vary, which do change under neutral evolutionary processes, which are not under selection. So the conservation comes from strong negative selection. So anything that should not change will be selected negatively or will be under negative selection so that it remains the same and does not change. Any region or any particular amino acid which is not really important for the whole process of the structure or binding with other molecules, those are now not under any selection pressures, and hence they can change. So there will be some minute differences in some of these amino acid regions which can actually change, which is what will give us an idea about how these bacterial species are changing or varying. And the size of the 16S rRNA gene is around 1500 base pairs, which can be sufficiently amplified by using very minimal efforts in any PCR reaction or any metagenomic sequencing reactions. And getting such a small gene is bioinformatically also very helpful because it does not require a lot of computational power, and especially in case of metagenomic analysis where we are sequencing the rRNA genes of thousands and thousands of microbial species, having a small gene which can be compactly assessed effectively is really helpful. And hence, this is also one of the reasons we select 16 as rRNA for the identification processes. And there are also like lot of 16S sequences available in the public databases. One such database is NCBI, which is National Center for Bioinformatics, and they have this data for sequences from lots and lots of organisms sequenced by different groups across the world and so on. And hence this particular gene has been sequenced so many times all over, and in case you want to compare your gene with something that has already been sequenced, or use the genetic information that you have and compare it to find what microbe it is or what bacterial strain it is, then it would be easy to just use that sequence and find the similar matching sequence from the database. Since this repository is very robust and it has a lot of sequences, sequencing a 16S rRNA is an added advantage. So 16S rRNA sequencing is suitable for identification of phenotypically aberrant, poorly described, or even rarely isolated strains. Like I said, there could be n number of microbial organisms, bacteria that cannot be cultured in the lab, and even in that case, if we are able to, or if we manage to get the 16S rRNA sequence, we will be able to match that with already existing sequences, or even for ones that are not well described, we will be able to classify them based on their sequence identities. And hence, classifying based on 16S rRNA is a better method to follow. And it can also be, like I said, it can also be used for organisms that cannot be cultured, and even novel pathogens that have never been isolated ever before. So if we get a sequence that does not match with anything in the database, then we can for sure say that this is a novel pathogen that has never been identified before. So this is one of the ways that we will identify or use the metagenomics to identify the different microbes that are present. But in any case, if we want to get an overall idea of the total number or the abundance of a particular bacteria, and also reduce them sequentially to go up to individual cell level, it is like most probable number technique is something that is often followed. It's called NPN technique, and this just basically is like a serial dilution method. It involves making dilutions in replicates of a particular sample in a very specific appropriate liquid growth medium and incubating them for growth. So there are multiple methods through which you can grow microbes under laboratory conditions. You can use agar plates, agar tubes. You can grow them in liquid culture. You can streak them on plates and grow them on solid surfaces, and so on. But MPN techniques involves serially diluting them in liquid media, and until we reach one viable cell in a particular tube or so on. So how is that done? Original sample. So here the example shown is original sample of pond water. This could be soil suspension or any environmental sample. From this main culture, it is serially diluted 10 ml and then 1 ml and then and 0.1 ml. So we first add 10 ml to each of these tubes, and from this we take 1 ml and add 1 ml to each of these tubes and zero point 0.1 ml and so on, and we'll make up the volume with whatever liquid media that we are taking, and we will incubate them at 37 degrees Celsius. So most ideal temperature for many of the microbial organisms is around 37 degrees Celsius, and 24 hours is a good time period that we can give to let the cells grow from the lag phase and then switch to exponential phase, and see them come to the saturation phase, or the dormant phase too. So here we incubate them and keep serially diluting them until we see zero positive clone in the given particular tube. So we should keep diluting them until we see any growth at all. So that is the principle or the basis of the MPN technique. So the MPN technique also gives an appropriate estimate, because depending on how many dilutions you do, you can back calculate and see how many number of cells could have possibly been there in the original sample that we collected. So this gives us an idea of the estimate of number of microorganisms in the sample. Even though we may not be able to get the abundance of a particular microorganism, we can get an overall estimate of the microbial load in a given environmental sample. And MPN is done in a highly selective media. Like I mentioned, the growth media that we are using in this case is supposed to be highly selective, especially if you want to estimate the number of a particular bacteria or a particular microbial organism. It is better to use one particular liquid media that will support the growth of just one microorganism, and the inoculum is serially diluted until we see any growth at all. That's the basic idea of the MPN technique. And most importantly, this technique can also be used for counting the number of anaerobic bacteria as well, because this technique particularly does not involve aeration or not using aeration. We can use this for anaerobic bacteria as well. We can grow these cells under anaerobic conditions and still get an idea about the total number of microbes. So we first saw how we can get an idea about the different types of microbes, and once we get that information from the 16s rRNA, we can use a particular liquid media to grow them and get an estimate of the total number of microbes that are present, or total number of bacteria of that particular strain that are present in a given sample. So we can either get an estimate of the total number of all microorganisms in a given sample, or by using a highly selective media, we can get the total number of a particular microorganism of interest. So this way we have an idea about the different types of microbes, or the identities of the microbes. So we get an idea about the species richness from understanding the 16s rRNA gene sequences. And by doing the MPN technique, we'd get an idea of abundance or species abundance, the abundance of a particular microorganism of interest. So we have classified them now and also looked at their abundance. So we have identified them, and the next step is to look at various techniques, how we can see whether there are live cells or dead cells. So this becomes important, especially when we're culturing them, to see how many of them are living, so that we can characterize their metabolic profile effectively. So there are various cell and nucleic acid staining techniques that are available for microbial staining. which can be used to differentiate live and dead cells. So even when we are looking at the growth curve, and if we want to know how many of them are live and how many of them are dying, and how actively the cells are dying in a particular culture, these cell staining techniques will be of help. So there are several staining dyes, fluorescent and non-fluorescent staining dyes that are available in the market. The very commonly used one is trypan blue, which stains the cell surfaces and the cell nuclei. So when we stain a live cell, and if the cell surface is intact, we'll see like a circle, which has the staining only in the borders of the cell, and inside the cell it will be empty, so it will be colorless, so it will be circles with empty dots inside. But in cases where the cells have already died, the trypan blue can pass through the cell surface, it can penetrate inside the cell wall, and it goes and stains the nucleus. So now what we see is a complete dot, like smaller complete dots that are filled with the blue color. So this will help us differentiate between live and dead cells, and this is a very straightforward and a very affordable staining technique as well. You don't need a fluorescent microscope or a fluorescent excitation emission spectrometers or anything. You can just see them by just adding the trypan blue and looking under a light microscope. We'll be able to differentiate between live and dead cells, and this is a very easier way of counting the viability of our cell culture as well. Similarly, HE staining, methyl green are all different cell staining techniques that are non-fluorescent. There are also fluorescent staining techniques. Like I said, DAPI is a very commonly used fluorescent staining technique. Similarly, PI is also an commonly used staining technique. So what is the principle of these different staining techniques, or what are all these different identification methods that we can use for differentiating live and dead cells? Is that, for instance, for DAPI, DAPI binds to single stranded DNAs. So it goes and intercalates with these single stranded DNA strands and at these different positions. So this is a positive stain, and it gets attracted to the negative charge of the DNA. And that's how, when we give the particular excitation, and if we look at the particular emission wavelength, we'll be able to see the DAPI staining for the nuclei. So DAPI basically stains the nuclei, and if it can penetrate through the cells and if it stains the nuclei, then we can know that the cell is dead. Similarly, propedium iodide (PI), which is a very commonly used stain again, binds to DNA and fluoresces under UV conditions. So this is a very similar mechanism as DAPI, where it penetrates inside the dead cells and it can stain the nucleus, whereas it can't stain the live cells. So it penetrates only cells with a damaged cell membrane. So once the cell dies, either by apoptosis or necrosis or any of these mechanisms, the cell membrane starts degrading, and that is a signature that the cell is dead. But in contrast, there are also several other staining dyes that are available currently that can be used for permanently staining cells that are specifically alive or dead, or even for live or even fixed cells can be stained. So Cyto 9 is such stain which can be specifically used for these different purposes, depending upon our need or depending upon our application. So now we have a growing culture. Even if we are not able to culture them, we already looked at the different microbes that could be present. So we now know the abundance, the species richness, and also how to keep the different cells alive in a culture, or how much of them are alive, whether they are viable or not, can be tested using these different techniques. They can either be visualized or they can be looked at under a microscope, and hence we'll be able to understand how viable our culture is. Why should we know the viability? Because only then we will be able to tweak the metabolic processes or perform more suite of experiments to now understand their characteristics, like their metabolic or ecological or physiological characteristics. So the next thing is characterization. So we already looked at couple of characterization techniques, like how to classify them based on their um, uh, the different metabolic processes, like what kind of sugar they consume, what kind of energy source, and so on. So we will also look at some of these characterization techniques in the next coming classes. But one thing that to just give an idea about how this characterization works is that now we have identified a bacterial strain, we know its identity, and now we are trying to grow it under anoxic conditions. And under anoxic conditions, which is anaerobic conditions, in the absence of oxygen, and in the presence of an organic carbon source like a sugar, and a nitrogen source like yeast, and in the absence of an electron acceptor, so that is under anoxic conditions, if we are able to produce alcohol and carbon dioxide, and if we are able to grow that particular microbe, this microbe is called as a fermentative microbe, and this process is called as fermentation. So this is how we metabolically characterize them. So we looked at what strain the bacteria is, and we gave different conditions for culturing them. We gave different carbon sources and nitrogen sources and electron acceptor sources, and we finally are now able to put them under different categories by characterizing their metabolic processes. So this is just one example of how we can classify them based on their metabolic process. So, to just brief, what we did in today's session is that we tried understanding how to first even know whether they can be culturable or not. We briefly looked at how the growth curve of an bacteria looks like, or a microbe looks like, what are all the viable cells and dead cells, what happens when they go back to regrowth or association, and so on, and what are all the properties that make cells culturable and unculturable. We also looked at the great plate anomaly, and how most of the microbes that are there in the environmental samples, or something that can't be cultured under laboratory conditions. We also looked in detail about 16s rRNA, how that is useful as an housekeeping marker and a gene that is reliable for sequencing, for identifying bacteria that cannot be cultured in the lab, and how the metagenomic information from 16s rRNA can be very helpful in the taxonomic classification of environmental microbial samples. We also looked at how we can perform NPN technique to understand the abundance of each of these different microbial organisms, even though we don't know the exact way to understand for the overall. We'll be able to specifically know the abundance of the different microbial organisms by specifically selecting the media that is required for growth of a particular bacterial strain, and so on. We also looked at how to differentiate live and dead cells, so that we can now know whether our culture is viable, whether it is actively growing or not. And with all of this information in hand, we saw a very brief about how to classify them based on their metabolic characteristics, and characterize their metabolic features. So, okay. Yeah, so this is what is there in overall of whatever's there in this week's lectures. So if you guys have more questions or any issues with solving any of the assignment questions, you can post it here. If there are any doubts from these sessions, you can ask those questions as well here. Any questions? So whoever has joined late can always go back and look at these videos. They'll be available on YouTube. So you can check the videos and see. If you don't understand something, you can always mail me. I can reply in the discussion forum.
May I know your mail ID, Sinji?
Yes. I can put it here in the chat folder.
Okay.
Yes, that's my mail ID. You can mail me if you have any questions.
Thank you.
Yeah, thank you, sir. Any other questions? Any like you?
Ma'am, I have a doubt.
Yes, yeah.
It's not. Ma'am, I have seen the recent significant development in biogas plants in India and everything. They are majorly based upon methane, and they come under green. मतलब the emissions or come as well as to reduce the carbon footprint in short. But in short, methane itself is a potent source for the CHGs, greenhouse gases. And
Yeah.
so, ma'am, how is it using methane by microbes? We use everything, processes and everything. So, is methane released into the environment, or it's just in the container chamber? It's just transferred into energy, and no emissions are made. So,
Yes.
there is no significant details about it.
Yes, so methane is like I think in the previous sessions we saw that methanogenesis is a process how methane can be taken in by microbes and can be converted into useful microbial products, secondary metabolic products. So methane has this, and methane is also something that's released from most of the normal bioorganic processes as well. So any decay of any organic matter releases methane. So, even though methane in higher quantities itself is a greenhouse gas and can have really serious impact on the environment, as compared to other gases like CFCs, like chlorofluorocarbons, which are highly poisonous and can have really bad effects on human health. Methane is something that can be converted into some other products by microbial action. So most of these industries which have a green certificate, which are working under the environmental norms, have to convert this methane into byproducts that are not harmful for the environment before they release it to the environment, or the methane levels have to be in a controlled way so that they can be absorbed by the environment without causing any serious effects to the atmosphere and so on. So as compared to other effluents that are highly harmful, methane is relatively better, and hence the new regulations allow methane to be an industrial exhaust.
Okay, ma'am. Uh,
Thank you. Any other questions?
Ma'am, what is microfluidics? I just heard this new term around. Could you share anything light on this regarding to the biotech?
Microfluidics could be anything to do with to assessment of these uh, smaller quantities. So especially environmental samples which are very smaller in amounts, microfluidics is a very it's for precise control of the release of the quantities of samples and so on. So this is used in many of the uh, machines like the ones used for transcriptomics, proteomics, and so on. Since the quantities available for working with is very less, they use microfluidic chambers and instruments that will release very less and precise volumes of samples that can be exactly assessed. So that's basically micro plus fluidics. So it's just precise control of movement of solutions or liquids under controlled conditions, especially for biological samples that are available in lesser quantities.
Okay, ma'am. So we are already using this all these things in biotech. Already we are manipulating the artificial environment for the microbes and everything. So microfluidics is just for smaller versions of nanomaterials and
Yes,
everything.
yes. For it's basically most of these processes that people used to do early on, the laboratory techniques like culturing and analysis used to happen in test tubes and such higher volumes. Since we don't get such high volumes of samples in most of the cases, things have changed, and now with the power that we have for analysis, we are able to analyze very little volume. So basically, it's just looking at very micro volumes of environmental samples or any biological sample for that instance. So it's just basically replicating that whole test tube based processes under micro or nano volumes.
No. Thank you, ma'am.
Thank you. Any more questions? Anything else that you guys want to ask or suggest? I hope all of you have registered for the final exam. And okay, Hitesh has a question. Example in genome sequencing microfluidics is used. Yeah, yeah. Yeah, that's why I said most of the omic techniques now involve using these microfluidic chips. So you can call like an entire lab works on a small chip controlled by microfluidics. It's used in fluorescent sorters like cell sorters and um, cell analyzers. It's used in NGS sequencing like Illumina platform uses microfluidics and so on. Any other questions from the existing assignments? Any issues? All right, if there are not any more questions, I think, okay, yeah, thanks, Hitesh. Then we can probably close the session for today. I'll save the recorded video version and I'll put it on YouTube. You guys can go back and check if there are any doubts, and you can always send me your suggestions for future sessions. Thank you so much for attending today's session. Thank you, everyone.
Thank you.
Thank you.